let's get started. I will introduce our two speakers today. They are both at the Open University in the UK. So we have Michael Macey. Michael is a senior lecturer at the School of Environment, Earth and Ecosystem Sciences at the Open University and investigates the microbial communities of extreme environments and their relevance to hypothetical extraterrestrial life. Maybe not so hypothetical, who knows, via using bioinformatics modeling and lab simulations. And then we have Ilieva, who recently got her PhD degree, congratulations, with the Open University Astrobiology Group, and has been investigating in the same line of research, so microbial communities of hypersaline Mars analog sites, and of course that's what we will hear about today. She previously obtained her a master of Bachelor of Science of Molecular Biology from the University of Manchester. And now, well, she is in between state, I guess, and looking for work and still helping, of course, in the lab, working in the lab and writing up papers, as we all have to do. So without further ado, I will hand over to our speakers.
Thank you. Okay, fantastic. So starting off with me stealing the laptop. So today's talk is going to be about the concept of exploring habitability through the study of hypersaline analog environments. There we go. So hypersaline environments, we're referring to any environment that has a sodium chloride concentration exceeding that of seawater, so greater than 0.6 molar. But as we can see from the examples on the right-hand side, there are many environments that in fact greatly exceed that concentration of salt. Now, hypersaline environments are found globally and can often have additional extreme parameters. So those can be fluctuations in temperature, extremes of pH, and they also typically are in environments where they will be hit by incredibly high levels of ultraviolet radiation. Why are we interested in hypersaline environments? Well, as a microbiologist, I'm interested in one aspect with the metabolic capabilities of the microbes that survive there. So the extremophiles, specifically halophiles, the salt-adapted organisms, will possess adaptations that enable them to overcome the extreme selection pressure, specifically of water wanting to move across a membrane to a more concentrated solution. This would result in the death of the cell. So to combat this, cells can take a salt-out approach, where they produce organic compounds called osmolytes, such as glycine betaine or DMSP, and that helps to balance the internal chemistry and prevent this movement of water. Or you have the salt-in approach, where you simply have the importation of salt ions and enzymes that can withstand the incredibly high salt concentrations and typically low pH that's associated with that. Building on the fact that these extremophiles have unique adaptations that we don't always see elsewhere on the tree of life, they therefore possess biosynthetic gene clusters and may potentially produce metabolites with relevance to societal applications, including as novel antimicrobial compounds. So shown here are a couple of examples of papers where members of the actinobacteria have been isolated from the Atacama Desert, and in our own research on a hypersaline brine spring in the north of the UK, we've also been able to identify similar natural products, but in gram-negative bacteria, so in a completely different bacterial phylum. Then, of course, the study of extreme environments is the study of life at its very limits. So in this simplified graph here, we have all culture-based life, so all life that we know about through being able to cultivate it in the lab, in the darker green, and in the lighter green, we have all of those organisms we know about the existence of thanks to sequencing-based technologies, but that we haven't been able to get into pure culture as of yet. But we know that there are some environments that exceed these specific parameters on Earth and are therefore uninhabited. But we also know that across the history of Mars as a planet, it may have had environments which had an overlap with these extreme limitations of microbial life as we know it on Earth. So we can develop our understanding as to whether life could have potentially once existed in some of these former environments. So when we're studying hypersaline environments in this perspective, we consider them analog environments. So an analog environment is a region that possesses a chemistry approximating that of a celestial body of interest. So if we consider the surface of Mars today, We know that there are pockets of salt. We know that it's an incredibly oxidizing and anaerobic environment, and it's exposed to high levels of both ultraviolet and cosmic radiation. But this, of course, brings up the important point that an analog is an analog for a specific region and at a specific point in the history of that celestial body. So this is the surface of Mars today. We know that this would be very distinct from the subsurface of Mars today, but also very different from what we believe Mars to have been like in the distant past, billions of years ago. There is extensive evidence that's been gathered by various missions to Mars that water was once present on the surface, although the existence of a former ocean is perhaps a bit more contentious. But water would have been present and would have been present in various salinities. And as this water desiccated and dried out, it would have become hypersaline as well. There would have been oxygen present in the atmosphere, although again, it's debated whether this would have been oxygen present at greater concentrations, or simply that there was more atmosphere and therefore more oxygen. And then lastly, there would have been shielding from some of the radiation in the atmosphere. So when we're considering analogues, we're considering analogues for these former Martian waters. What can we learn by studying analogues? Well, we can gain insight into many different things, but specifically we can gain an understanding with regard to the metabolic viability of organisms that can withstand that chemical environment, the ecology therein, the microenvironments and how we get variation in what we would perhaps otherwise consider a monolithic environment, and the potential remnants of life that may be left behind. So let's walk through these in a bit more detail. In terms of metabolic viability, we're referring to metabolisms that provide sufficient energy to enable the replication of cells, so persistent, sustained growth in addition to survival. So shown here, we have a redox tower, which shows the different potential electron acceptors, terminal electron acceptors at the end of a metabolic chain. So at the very top, we have oxygen with aerobic metabolisms being incredibly highly energy yielding. And then for the rest of the redox tower, we have the subdivisions of different anaerobic electron acceptors. And the way that this would impact the community, if we look at these illustrative but representative of real species sulfur oxidizing microbes, you can perform the oxidation of reduced sulfur species with a variety of sulfur species and a variety of terminal electron acceptors, either in an aerobic or anaerobic fashion using oxygen or nitrate respectively. So dependent on the availability of these substrates and the relative abundance of these, that then means you would have selection for some specific variants and some specific metabolisms over others. Now, ecological principles and their influence on viability and community assembly is also important to consider in our concept of habitability. In an environment, if it's potentially inhabitable, the second you have a single cell, you have ecology because you have an organism interacting with its environment. If that cell divides, you then have a population. And if you then have any variation that deviates this monoculture from being completely identical, then you have an ecosystem. And we can expand some of these ideas of ecological theory to microbiology and astrobiology. So shown here, we have Darwin's finches, which is a famous example of an ecological niche. So the match of a species to a specific environmental condition. So they observed that there were multiple finches on the islands, but they were able to coexist because they had adaptations that gave them slightly different niches, some being able to crack open nuts and others being able to harvest insects more effectively. Now, if we go back to our example of sulfur oxidizing microbes, it's not quite as simple as reduced sulfur all the way to sulfates. There are many different enzymatic systems, and these have different preferences and requirements for pH, for cofactors within the cell, for different availabilities of metal ions. And this can mean that you can get simultaneous occurrence of seemingly overlapping metabolisms, but you can also get competitive exclusion of some of these because they don't have a sufficiently different niche and they vary in their competitive efficiency. And there's the concept of the keystone species. So here's the keystone of the arch, which is the keystone that holds the arch up. It's an organism that has a large effect on its natural environment. So in a field with grass, the keystone species would be the grass itself. It's pumping the carbon into that ecosystem. Doesn't necessarily have to be the most abundant, but it's inarguably having a large impact. And in an environment that's dominated by sulfur oxidizing bacteria, that is carbon poor, in this same way, the autotrophs, the microbes fixing carbon dioxide from the atmosphere, like the plants, would effectively be acting as a carbon pump. And if you were to remove that function, perhaps the ecosystem itself would not be able to sustain. And then lastly, in terms of ecology, there's ecological drift. in microbiology, it's always tempting to consider selection as the sole key factor because, of course, it's an extreme environment, so the microbes have to be withstanding the selective pressure. But random fluctuations occur. Cells die and cells divide. And owing to just shifts in numbers over time, you can get shifts in community structure occurring in the absence of any specific selective factor, but owing solely to just simple random fluctuations. Now, if we also consider microenvironments and how stratification within a selection gradient can occur, here we have the White Desert or the Great Rann of Kutch in India. Now, across this environment, there are many different microenvironments. So if we were to take a core sample, we have the microbes on top of, inside the salt crystals, below the salt, and then separated with depth. But also we can see at the sides, we have encroachment of plant life onto these salt crusts, which will also be providing other materials. So there are many different environments within this one specific extreme. And then there are, of course, putative biosignatures. Now, we know that there are microbes within these salt crusts. So if we take our core sample, we can analyze them not only with a view on what is the microbiology of this site, what's their metabolic diversity, we can gain an understanding with regard to are there any geochemical signatures we can detect as a consequence of this. And referring back to the Europlanet Transnational Access Scheme, thanks to this and researchers at Isotoptech in Hungary, which is one of the partner organizations, we were able to get isotopic analysis of both this core and many other cores collected from a diverse array of analog environments. We were able to show that there was this depletion of lighter sulfur across the depth of the core, with microbes typically preferring lighter isotopes because it's more energetically efficient. And if we then turn to the mineralogy of the environment and any potential inorganic biosignatures formed as a direct consequence of the metabolisms of these organisms using the SEM analysis suite at the Open University, we were able to identify that all of the sulfur within the core is now reduced, which is very likely a consequence of metabolic activity. We can then further analyze this for if there are any biosignatures that persist detectable at the mineralogical level. Overall, all of these things feed into the generation of hypotheses. And we've been very fortunate to work both through Europlanet and with our network of collaborators with a diverse array of analog environments. We're going to hear more about some of the sites in Spain today, but as you can see, there's an incredible diversity of these hypersaline sites across the world. So then the question is, if we can gain this insight into potential habitability on other worlds by studying an analog environment, what's the benefit of studying multiple analog environments? Well, no two analogues are identical, and this is critical because minor variation in conditions can have major impacts on the viability of distinct metabolisms. So shown here, we have a graphical representation of fitness. Now, fitness is relative on the environment. And so if you have two analog sites, one of them has a higher concentration of magnesium salts relative to another, you would expect the landscape and where these peaks representing fitter organisms would shift significantly. So this is important with a view on the fact that we, by studying analogues, are looking at an approximation of an approximation. We are studying environments on Earth that have been exposed to oxygen and the presence of life in order to try and gain an understanding of sites that we are building an understanding of through remote observation and rovers and landers on the surface of that planet, and then using modeling to scale this back through time to try and gain an understanding of what those environments may have been like. So we consider the potential analog space of Mars, this orange sphere, and the full spheres, the potential perspective that the analog environments give us into this. It's a way of exploring this potential physicochemical space. Now, I'm very happy to pass the computer over to my former student and now Dr. Ilieva to talk us through her detailed research on the La Mancha region.
Thank you very much, Michael. Today, I would like to tell you about the La Mancha region in Spain as an analog for the sulfate-rich microenvironments on Mars that we think have existed several billion years ago, and especially the early Hesperian period. The La Mancha region in Spain is located in central Spain, and it's an arid region. The summers, there's very low precipitation, really high evaporation rates, and during the winters, there is a lot more precipitation. So the 4 study sites that I investigated for my PhD project are shown here. They're relatively close to each other. So for example, between Igúes and Longar, there is about a 25 to 30 minute car ride. And also, interesting fact, this is the same region that is mentioned in the really famous novel Don Quixote de la Mancha. So while we were sampling some of the sites, we could see the windmills in the background, which personally was a joy for me. Having a bit of a closer look at the 4 study sites, Yaguas, Penahueca, Quero, and Lungar, you can immediately see from the pictures that even though they are close to each other and they're in the same geological context, they are very different. Going first at Yaguas, we see that there are only small-scale deposits of salt on the surface of the lagoon compared to some of the other sites. At Penahueca, we saw these polygonal features that had salt deposits at the borders and sediment in the middle of the polygons, so we sampled both. At Quero, we also had polygonal features, however, they were different. The whole surface of Quero was covered by salt deposits, so in the center of those features, you had much thicker salt that was raised towards the borders, and we could see sediment peeking through. And finally, at Lungar, we had more sporadic salt deposits across the surface of the lagoon. So as Michael mentioned earlier, a Mars analog site, Mars today is very different from Mars in the past, but also the subsurface of Mars today is different from the surface that we have so extensively studied. So with that regard, we collected salt deposits from the surface, which have also been identified on Mars, such as sulfate salts and some halides, but I'll go into that a bit later on. We also collected sediment cores, and we found biofilms at 2 of the sites. So at Yaguas here in figure A, we saw subsurface biofilm about 2 centimeters under the surface of the lagoon. And at B, at Lungar, we saw surficial biofilm. So there is a bit of water because we sampled in November 2022 at the end of the dry season. And we were actually very, very, very fortunate to be hosted by Dr. Felipe Gomez from the Center for Astrobiology in Spain that took us to those sites and helped us sample them. So it was me, myself, Michael, and Felipe. To reiterate, the 3 sample types that we collected and I analyzed for my PhD were salt crust deposits, sediments, and biofilms. Some of the sediments had a really striking stratification that was immediately visible in the laboratory as soon as we opened the samples. For example, here on the top image in the middle, I've shown a sediment core from Penahueca, and I've divided it in sections. You can see that at the top of the sediment, between 0 and 5 centimeters, you can only see thick sediment, whereas that changes with depth. At 5 to 10 centimeters, we see mixture of salt and sediment, and at 10 to 20 centimeters, we see entirely salt deposits. And then at 20 to 30, again, another change in color as well. We see clay-like sticky mud with orange color. So we were very interested to begin this analysis. For the geochemical analysis, we performed chemical and mineralogical analysis, and I've shown the data here now separating the center and border sections for Quero and Penahueca, but I'll just talk you through several key points. First, Yaguas sediments have the lowest salinity. So A means the top of the sediment, and E or D is at the bottom of the sediment. Again, we did not see much variation. It was still saline, but not in comparison with, for example, Penahueca, where we saw a lot more salt-associated ions and elements, such as sodium and chloride, as well as sulfates. At Lungar, we had some changes with depth, but again, they were much more uniform, such as Yaguas compared to Penahueca. And at Quero as well, we saw a lot more uniformity in chemical composition with depth. And then the mineralogy analysis kind of confirmed, obviously, the chemical data and told us what these differences are between the cores. The first key thing is that Yaguas and Lungar, as well as Quero, are very gypsum-rich. So gypsum comprises over 70% of the mineral abundance of these sites. At Quero, we see some differences between the center sediment that was covered in thick salt crust and the border, and we see some magnesium sulfate minerals as well as bloodite. At Penahueca, the story is very different. So with Penahueca, we see the same stratification that we saw in the chemical and just with our eyes as well as on the chemical data. At the top of the core, we had a lot more halite, as well as darkite. And then as we're progressing through, the mineralogy changes. So in section B, where we had mixed sediment and salt crystals, we see that we have gypsum as well as bloodite crystals. So these are sodium magnesium sulfates. It's called a double salt. And we have different magnesium sulfates in different hydration phases. Then section C, which was entirely comprised of salt crystals, we identified those as the double salt bloodite. And then at the bottom, we identified the orange clay-like sediment as clay, muscovite, and then as well we had some quartz, calcite, and dolomite. So then we performed microbiology analysis. For this, I helped sequence the samples for 16S RNA gene amplicon sequencing, and we also send them off for shotgun metagenomics. So first, I'm going to talk through the amplicon sequencing, which helped us identify the taxonomic composition of the samples. There is quite a lot of data here, but focusing on a few key points again, is that I performed hierarchical clustering analysis of the communities, which essentially groups communities closer together if they're more similar in taxonomic composition and abundance of these taxa. And what I saw is that all of the Penahueca salt and sediment communities grouped together with the salts and biofilms from the other three sites, forming the first cluster, whereas the second cluster was entirely composed of sediment samples from Yaguas, Acero, and Lungar. We see halobacteriota sequences, which are halophilic archaea, that is completely expected at these highly saline sites, as well as sulfate-reducing bacteria-related sequences here in the bright blue. And one of the key differences between the two clusters was the much higher abundance of what they were formerly called acetothermia sequences in the dark blue, or Candidatus bipolariculata. And in the Penahueca samples, we see here with purple a lot more nanohaloarchaeota, which are these small halophilic archaea. So the taxonomic profile absolutely fitted with what we've seen from the mineralogical and chemical data, but also now I had two lines of evidence telling me that Penahueca seems different compared to the other three sites in terms of the geochemical environment and the microbial communities. So in order to understand what is happening a bit better, I performed ordination analysis or unconstrained analysis, and this is where I created these two non-metric dimensional scaling plots, where essentially they group communities that are alike closer to each other. So in the first plot, communities were essentially grouped together in regard with Bray-Curtis dissimilarity, which takes into account taxonomic composition and abundance. So a bit similar to the hierarchical clustering. And that showed the same tendencies like the Penahueca communities grouping closer together away from everything else. Also, for this type of analysis, I added the environmental variables post hoc onto the plot and performed statistical analyses to identify which environmental variables are linked to the communities. And this is where we have the asterisks above certain parameters. For the weighted unifrag distances on the plot number B, very similar kind of concept, but we take into account phylogenetic relatedness as well as similarity in taxonomy. And we see that, again, Penahueca are slightly away and they are linked to the concentration of sodium. To confirm these findings, I also performed constraint analysis. This is called distance-based redundancy analysis, where I essentially into the model, I used the microbial data as well as the environmental data, again, basing on the same Bray-Curtis similarity and weighted unifrag. And those gave us the absolutely same conclusions that the Penahueca communities were different to the others, and they were affected significantly by the concentrations of sodium as well as chloride, which makes sense because we found a lot higher concentration of these elements and ions at Penahueca compared to the other sites. However, as Michael mentioned, ecology is key for studying analog environments and when thinking about life in general. So the next type of analysis I performed on the data is ecological no modeling, where essentially I use the phylogeny to infer ecology. There's one underlying assumption for this type of analysis that's quite key to remember, and this is that species that occupy similar niches are more closely related than expected by chance. So what does that mean? We first need to define a few things. What is a species? In this case, species is an ASV or an amplicon sequence. We talk about the sequences from the amplicon sequencing analysis. And then niche, I defined niche as the parameters that significantly correlated with the structure of the microbial communities from the previous statistical analysis I performed. So these are pH and the concentrations of sodium, magnesium, calcium, potassium, silicon, phosphorus, chloride, and sulfate. And then I calculated the optimal niche for each species. And I did this by calculating the abundance weighted mean, or what are essentially the optimum pH and concentration of each of these things for each ASV. And secondly, I had to also identify the phylogenetic relatedness between each species, and this was done by creating a maximum likelihood phylogenetic tree. So in order to satisfy this assumption of the analysis that species occupying similar niches are more closely related, I then performed mantle correlation statistical analysis, which I'm not going to go into the details, but confirmed that this is true for my data. So now we can actually go into the ecological no modeling analysis after we have satisfied the underlying assumption. There's two parts to this analysis. This is a type of beta diversity metric where we're comparing two communities, but multiple times. So for the part one of the analysis, I wanted to understand which communities are more closely related to each other than expected by chance and which are less. And then for part two, I looked into the similarity in taxonomic composition, but I'll go into a bit more detail about each part now. So in part one, the phylogenetic relatedness. So all of this was done by a package in R. I didn't do any of the maths or anything like that by myself, but this is kind of just very, very superficial walkthrough through the process of what those packages are actually doing. And when thinking about community A and community B, for example, two communities, first we need to identify how related they are in terms of phylogenetic distance. So what we do is we calculate this metric called beta mean nearest taxon distance, which it's beta MNTD. It doesn't matter what it means, but that gives us a number. So essentially compares two communities and gives us a number how closely related they are. Then for the no part of the analysis, we perform a randomization of all species in these communities along the phylogenetic tree, and we redo the analysis. In my case, I rerun it for 10,000 times. So that gives us a distribution here at step 3 of what would be expected under normal conditions. And then essentially we compare the observed with the distribution that we have calculated. And here at step 4, that deviation tells us whether two communities are more or less related than expected by chance. So if these communities are more related than expected by chance, so less than -2 here on the plot, they're affected by homogeneous selection, which means that their niches are actively exerting selection on those microbes. If those communities are less related than expected by chance, so here after more than +2 for this index, they are affected by variable selection. And variable selection would work in cases if, for example, you compare a community from a really hot environment with a community from a really cold environment, you would expect that these two communities are affected by variable selection because you have two very different variables, temperature in terms of temperature, acting on those communities. So I've done this for all community pairs in my analysis from all of the sites. And those communities that fall within the distribution then go on to the second part of analysis, where to further tease out the stochastic processes that affect them. I look at the similarity in taxonomic composition. So this is done by another metric, and essentially it's another Bray-Curtis distance. But what we are doing there is that if species have less number of species that they share if two communities have less number of species that they share together, they are influenced by dispersal limitation, essentially meaning that species from community A cannot travel to community B, so there's no exchange. If two communities have more species similar than expected by chance, then they are affected by homogenizing dispersal, which means that the two communities can exchange species. And finally, for those that fall within the distribution for this metric, and they are not more closely related or less, they are under the influence of ecological drift that Michael introduced earlier, where we had random live death events. So after all this complicated setup, the output of the analysis is actually very simple and straightforward. Here in figure A, I've compared the communities within each of the site with each other. And I saw very clearly for Yaguas, Kero, and Longar, their communities are mainly affected by stochastic processes in the blue shades of blue. So we see that homogenizing dispersal and ecological drift are among the two dominant ones. So again, very expected for communities at these sites. And it means that species can travel, for example, up and down the sediment column. This can be caused by underwater upwelling up and down the sediments, which would transport the species between communities. And for Penahueca, we see that the things are very different. So the microbial communities are mainly affected by homogeneous selection, where the environment is exerting strong selection on their species. So also we can think of it as Yaguas, Kero, and Longar. Their communities are very used to the extreme and oligotrophic environment, whereas those at Penahueca are continuously being stressed by the environment and changes in the environment. Because of the chemical mineralogical data and the statistical analysis beforehand, this gives us evidence that it is the salinity, the fluctuating salinity, and the very limited water availability at Penahueca that is causing this stress on the microbial communities. And to further confirm these findings, here in figure B, I compared communities across sites. So this is where I compared, for example, communities from Yaguas to Longar to Kero. And we see that those communities are affected by dispersal limitation and ecological drift mainly, which means that, yes, communities from species from Yaguas would be very difficult for them to travel all the way to Kero and vice versa. And when we add Penahueca to this mix and we compare those communities, we see that variable selection here is the main driving force. As I said, variable selection you would expect to act on two communities that live in very different environments. So now, the next thing we also did, we had a quite good idea of what is happening to these communities, is we wanted to see how they survive in these extreme salinities. So for that, we sent the samples off for shotgun metagenomic sequencing, and this is just a snapshot of the data. So essentially on this figure, you can see all the metagenomes that we sent from all the 4 sites. And each line represents a complete metabolic pathway linked to living at high salinities. Without going into too much detail, it's important to note that we found strategies for both surviving at salt in and salt out. We found pathways for both salt in and salt out strategies, which is, again, very expected for communities at high salinities, especially fluctuating salinities. And finally, I will just quickly talk a bit about biogeochemical cycling, because this is highly relevant to hypothetical life on Mars. And I will focus on each metabolism separately. First, sulfur cycling. Sulfur is extremely important on Mars. Sulfur is very abundant on Mars, and we've detected sulfur phases, such as oxidized and reduced, that life could potentially gain energy from. So with that extent, I also looked for those sulfur cycling genes in my site, and I found genes for sulfate reduction, So this is where microbes use sulfate as a terminal electron acceptor. So it's an anaerobic type of metabolism. And genes for sulfur oxidation that Michael explained earlier, it's a type of chemotrophic metabolism where microbes extract energy from reduced sulfur minerals. And we can see that it's those genes were detected at all sites, but a bit less at Penahueca, where the high salinity could potentially inhibit that metabolic strategy. But at some sites, I'm just going to highlight here Yaguas, we have a really nice stratification between sulfur oxidation between 0 and 20 centimeter depth and sulfate reduction at the bottom. So this is an example of sulfur cycling in the sediment column. For nitrogen-related genes were a lot less prominent here at the site, which actually fits quite well with Mars. The nitrogen cycle is less important, and also it is inhibited at high salinities. So we have some genes linked to identification, but not much. Other sources of energy for this oligotrophic environment is hydrogen. So these hydrogenases are enzymes that allow the conversion of hydrogen to other states in the extraction of electrons. Hydrogen is a bit less relevant to Mars. However, there has been work that's shown that serpentinization reactions that could have potentially occurred on Mars could provide hydrogen that can then be used by microbes for energy. And finally, one carbon compound. Again, carbon monoxide and carbon dioxide are most relevant for Mars, of course. And we saw evidence for their utilization here at the sites in La Mancha. Formate and methylamine a bit less because they have not been detected on Mars, but here on Earth, they are quite important energy sources for microbes. So just to summarize, through this work, I've performed a very detailed characterization of 4 lagoons in the La Mancha project, in the La Mancha region. And all of the data pinpointed that Laguna de Penahueca is slightly drier than the other lagoons and might be drying up completely. Interestingly, there is another lagoon which is features quite prominently in astrobiology studies. This is Laguna de Tires, which some of you might have heard about. That's only 3 kilometers away, and it has completely desiccated in 2015, and it has been identified as a temporal analog for desiccation on Mars and the effect of putative life. So Penahueca is very close to that, and a recent study also came out to say that the underground water table at Tires and Penahueca is much lower compared to previous years, which likely contributed to the complete desiccation of Tires. The same process might be happening at Penahueca. Therefore, Europlanet were very generous to give us another round of funding so we can resample those sites in 2025. It was the original team of myself, Felipe, again, very generously joined us as well, and Michael, as well as our first year student, Rita, and she actually took those stunning images of salt deposits here in the middle. So now we're in the process of analyzing those samples and comparing them to the 2022 dataset. Thank you very much.
Thank you so much, guys, for this wonderful talk. Very interesting research indeed. Let's open the floor for questions. I see there's already one question here from Amy. There is, right? From Amy Quinton. From the biogeochemical cycling section. Could you explain how the normalization counts were calculated per sample for the presence of each gene involved in a certain metabolic process?
Yes, absolutely. That's a great question. So I used a variance stabilizing transformation, which is, I can't remember the name of the R package, but it is there. And it's been identified as a good way of normalizing the gene counts.
Okay, I hope that answers the question. Okay, so any other questions at this point from the audience? You can also speak your question if you want to, just instead of writing it in the chat or in the Q&A, if you want to speak, that's also fine. Do we have another one? No.
Not just yet, but that's okay.
I thought I saw something pop up, but no. Okay. So
Maybe
you
a very clear presentation.
Yeah, so you really focused on Spain, but you also mentioned the UK, in the north of the UK. There, Michael, there are some areas. Did you go? Well, why did you choose those specific sites in Spain and not other sites at this point?
So Spain was selected because you do get this wonderful variation within like quite a close site. It was also a project that was developed towards the tail end of the COVID lockdowns. So when we were designing the project, it had to be with a view on somewhere that would be reachable. There were travel arrangements with, we had collaborators in the host nation that absolute worst case scenario, all travel is banned again. Samples would still be potentially accessible, or there might be archived materials we could work with. So the site in Droitwich is somewhere we've come across more recently. So there used to be, since the Roman times, they've harvested salt from the brines underneath the town. That is now currently being restored, and there's salt baths being rebuilt, but that's more of an underground well. So it's still a hypersaline brine, but it's less relevant for the specific type of environment that we were interested in for Veli's project.
Okay, fair enough. Any other questions? Not yet. I don't see any. So what's up next because you now have all these results. You're writing them up, I guess. I mean, you already had it in your PhD, of course, your PhD. So what's the next step?
So actually, we managed to secure some more funding to do sequencing of the new samples. And the issue that we're struggling is having people and money and time to actually analyze those samples. So every little helps. So Michael has been really proactive in identifying additional funding that we can utilize for those samples. But I think it will be a great data set to compare between, even if it's like only 3 years apart, because as I mentioned in Tires, that completely has dried up. They, in one of the papers that they published in Nature a few years back, the lab from the Center for Astrobiology in Madrid analyzed sequences from like 10 years ago and then 5 years ago. So they had a really nice kind of data set. So this is similar to what we're trying to do as well. And it's quite informative for as well the local people. There is conservation efforts undergoing in this region, because previously it used to be essentially used very extensively for agriculture and irrigation. So now the government is trying to make so that these environments are preserved, those wetlands, because they're quite unique and they attract a lot of bird viewers. I think we scared some bird viewers when we were sampling our sediments earlier last year.
Okay, excellent. I think there's another question, Callum, right?
Yes, we do. So from Matilda asks, did you observe any phototrophs, e.g. cyanobacteria for oxygenic photosynthesis or green sulfur bacteria for anoxygenic photosynthesis? I hope I've pronounced all of those words correctly, in the biofilms you observe.
Thank you, Matilda, for the question. So very interestingly, there were some cyanobacteria in the biofilms. However, algae was the dominant kind of microorganism doing the photosynthesis in those biofilms. So we actually had to troubleshoot using algal databases to identify the sequences in our samples. But yeah, we thought it would be mainly cyanobacteria, but it was actually algae. Thank you.
Okay. I'm just wondering about the effect of the weather. So now, of course, this area is very arid, as you said. So we have these very salty areas, very arid. Okay, but now it has rained a lot. We know in this area as well. How does that affect? And do you have any information about that, or?
Oh, yeah.
So in terms of what we would detect at the DNA level for the samples that we took, because like the end of the dry season happened quite literally whilst we were taking some sample cores, and the heavens opened and we got soaked. But in terms of the microbes within the salt crystals that don't immediately go into solution, in terms of the DNA of the cells within the core, we wouldn't expect there to be major changes. DNA is comparatively stable for an organic molecule, is very stable. So in that regard, it's useful. There's also then the caveat, of course, of we may now be detecting cells that have potentially lysed. But we sampled the same site before and after the rainfall and processed those with 16S sequencing. And I don't believe there was any major sequence variation we detected. The main thing we had to avoid temptation with was, we can sample this water and this will be water from this site. But if it's been on the ground for 5, it's still rainwater. It's just salty rainwater. So anything we detected wouldn't necessarily be pertinent to our research questions at the time.
Okay. So there's no change in the whole community of microbes as such.
Nothing that we detected in the immediacy. What we might expect between years is, as it gets increasingly desiccated, that may have significant effects on the microbial community. So the algae we would expect to potentially struggle a lot more in the absence of the rain. And what we can do using the amplicon sequence variant approach that was used for the 2022 samples is you can identify if it's literally identical sequences. So as in, is it genuinely representatives of exactly the same taxonomic group that we're detecting between time, which I think is the best approach that we could take for trying to track that.
Okay.
And also, I was thinking about Penahueca, if we remember that core, how it was stratified with sediment at the top. So the top obviously is exposed a lot more to precipitation. And
is that the picture with the horizontal?
Yeah, absolutely.
Yeah.
So at the top, you'd expect maybe a bit more changes as rainwater directly obviously goes in contact with these communities and the sediments. However, up to 20 centimeters depth, there was these pristine salt crystals that were, they were untouched by the water, even if there was water penetrating. So again, it depends how far the water penetrates. And as I said, the groundwater table that has decreased in the recent years. So if you don't have that recharge from underneath the communities, that means at depth, they're probably pretty similar. And you might see a bit more variation on the top after rain. But again, as Michael said, we have done some comparisons.
Yeah. So this I'm just so this is on the Earth analog side. How do we think about going to another planet doing this kind of stuff? It's in the future, of course, but
well, I think both of us are quite excited about the Rosalind Franklin mission with the 2-meter drill, because as Michael really eloquently said in his talk, we have studied the surface a lot of Mars, but we have not really studied the subsurface. So even if we are able to, go 20, 30, 40 centimeters below the Martian surface, we might, we don't know what we're going to find there. So I think subsurface life is definitely very interesting. If you'd like to add anything.
Yeah, I think it's also interesting with a view on, obviously, when we're trying to take the results from analog environments and apply them to another planet, you run into the issue of single origin of life event or multiple origin of life events. I think taking the focus on metabolisms and interactions, if you consider like a finite number of redox pairs and the number of available potential electron donors and acceptors that exist on Mars now and may have existed in the past, I think it's interesting to study these environments with a view on, can we detect any changes that are exclusive to there being biology here that would potentially be able to withstand if there hasn't been any biology in the intervening billions of years.
Okay, good prospects. There's another question. No,
we do have. We do have another one from an
questions.
Anonymous attendee. So to follow up with Matilda replies, "Thanks a lot for the answer." Sorry, one more question.
Two more questions.
You're
There are
right.
two more questions.
Do you have any idea how algae / cyano protect themselves from the high UV radiation, as too many photons can also damage the photosystems, where the algae / cyano on top of the sediment or biofilm or below other species?
I think algae are a bit more sturdy than cyanobacteria in some cases, or
they have more repair mechanisms than
Yeah.
cyanobacteria. If
Yeah.
a single cyanobacterial cell gets damaged and dies, then it's dead. Whereas with algae, it can have cells die and then be repaired. It's very similar to what is seen in organisms like Deinococcus radiodurans. It's not that it's not being damaged, it's that it's making the repair proteins very, very quickly and en masse and repairing that damage. So I think being multi-celled is to its benefit in being able to withstand that damage. In terms of the localization?
Yes, so cyanobacteria, they were towards the top of the sediment column, and as I said, the biofilm, actually, there were some, in the subsurface biofilm at Ilieva, there were some cyanobacterial sequences, but there was a lot of halomonas, which was a bit surprising as well. So again, this is part of ongoing work because biofilms were not a huge focus of my PhD with regard to Mars, but it is on the publication list, to-do list, to actually describe those two biofilms a bit more. And also, was there a different part of the question? I think we started. Thank you, Matilda.
Martin, will you take the next one?
Sure. So there's two more questions now. There's an anonymous attendee who asks, how localized does the rainwater accumulate? Is it small isolated puddles, or also flow from one side from other sites that might also transport microbes between sampling sites and kind of homogenize the uppermost communities, while the subsurface ones are more distinct, I guess?
So the 4 lagoons are characterized as endorheic basins. So the main kind of input is either underground or as precipitation, but there are no immediate sites in the vicinity where rainwater could have precipitated. I think we have to think about air or like wind as a medium of transporting microbes, so that can affect like the top layers, for example.
Yeah, no, that's definitely a factor. It's always interesting to consider input of materials. I know that some extreme environments you often detect like unusual sequences, things that aren't extremophilic. Some of those will be contaminating sequences as are present in DNA extraction kits, but some of them could be something as simple as a bird landed there and ate something, pooed, and then left. To try and minimize the impact of things like that. So birds living in their natural environment and not caring about astrobiologists. We also were very specific to try and pick sites that were slightly further in, further away from any potential exogenous materials that might get put in. We're less of a factor here, but I have been to sites that there's quite happy flamingo flocks just moving around and then doing what birds do. The presence of animals often can complicate things as an input of materials as well.
Thank you. Next question comes from Nicole Ruiz, who asks, since there are many types of Mars analog on Earth, depending on the objective or focus, where is the homogeneous one in the solar system, specifically on Mars?
I'm afraid I'm not sure I completely followed the question.
Okay. Nicole, could you maybe in the chat or in the Q&A, could you expand on your question to make it a bit more clear for us? In the meantime, let's move on to our next question, which is from Traz. Nice to see you, Traz, who asks, thank you for this very interesting and informative webinar. I noticed that you identified a clay mineral in your samples. Have you attempted to determine the specific type of clay mineral present? Thanks.
Thank you for the question. So we know that it's muscovite, the clay. However, I have not done any further characterization of that muscovite.
Yeah, clays are a problem with DNA extraction as well. Like
Yeah,
they are very good at binding to compounds like DNA that have a charge. So yeah, it's surprisingly much harder than the salts to do the DNA extractions from.
Thank you both. Nicole, perhaps to expand on that question, as we are at the end of our time now, Michael and Veli, is Nicole, is it possible for you to be contacted following the session if there's more conversation to be had? How can we do that?
Yeah. Please feel free to reach out. So my Open University email address is just my name, so michael.macey@open.ac.uk, and yeah, happy to field any further questions.
Yeah, same here.
Perfect.
But my name's a bit more complicated and long, but it's velislava.ilieva@open.ac.uk.
Thank you both. Okay, sounds like that's cool with Nicole, so we can make sure that question does get answered and we still keep to our time. Thank you both for the talks. Thank you for asking your questions, and thank you to all our participants for dialing in and joining us today. Anything you'd like to say, Martin, before we close?
Nope, all good. So thank you very much, of course, and I guess see you next time.
Thank you so much.
Keep an eye out for the recording of this one. We'll see you all in 2 weeks for our next webinar. Until then, see you soon.